Network Journal Clubs. If no one objects, we're going to record this again and put it on the website. We're actually getting a decent number of views on there, I think, from people who have other meetings going on or just can't make it. So if you have any objections to being recorded, please say so now. Okay, no issues.
Cool.
So as Cherie mentioned, Anastasia is going to be running questions. So if you click on the bottom of your screen and enter questions, she can kind of curate those as we go. And so for now, I'll just introduce Matthew Kang, who is a PhD student under Larry Chamley in the Department of Obstetrics and Gynecology at the University of Auckland. And Matt, go ahead, take it away.
Cool. Thank you very much, Colin. Right. So for my journal club today, I chose this paper, The Mechanisms of Nuclear Content Loading into Exosomes. I thought this was a very interesting paper, just looking at the title. It turned out to be quite a long paper, so I had to truncate quite a bit out of it. So I did put in the most important parts that I think were the most important. So yeah, I'll begin. So the EV field is in its infancy at the moment, and we still don't know quite. We don't know. Well, there's still much more to learn about the sorting of cargo into EVs. We do have some idea of RNA and protein sorting, with, for example, ubiquitinated protein by CD63, such and such, but we don't really know much about the DNA cargo content sorting into EVs at all. So this paper provides that idea of how EV DNA can be packaged into these exosomes. So just a background. So intraluminal vesicles, ILVs, are generated in early endosomes, and they are stored in multivesicular bodies. So when these multivesicular bodies are guided towards the plasma membrane, they fuse with the plasma membrane to be released as exosomes. So the exosomes can contain diverse cargoes. For example, RNA, microRNA, mRNA proteins have been quite well researched. However, the DNA content of exosomes is still, it's still in its infancy. We don't know quite a lot about it. But more recent papers are showing up that genomic DNA is indeed present, as well as its nuclear proteins in exosomes, and this is especially the case for cancer cell-derived exosomes. So the thing about DNA is that, paradoxically, genomic DNA is mainly confined to the cell nucleus and does not normally interact with the multivesicular bodies in the cytoplasm. So, yeah, the mechanism behind how that genomic DNA is being transferred into that exosomes is quite a mystery. So what they propose here is something called micronuclei. So micronuclei are cytoplasmic structures that are enveloped by a nuclear membrane, and they contain chromosomes. So they are generated when the cell nucleus fails to properly segregate nuclear material during mitosis, but they can also be, this missegregation can also be artificially induced by treatment with artificial DNA damaging agents, such as radiation and also anti-cancer agents, which can result in chromosome fragmentation and subsequently genomic instability. So the presence of the micronuclei can serve as a surrogate marker for the genomic instability. So in their paper, they hypothesized that the genomic DNA that is sorted into exosomes are derived from nuclear material in micronuclei. So the first part of their result was looking at the cell line of choice and also the characterization of their exosome. So using a cancer genome atlas, they determined, they looked at different tumor types. I think it was 25. I think it was around 25 different tumor types. And they looked for one that had the highest ploidy, i.e. Hence genomic instability. And they came across the ovarian cancer cell lines. And they ended up with the ovarian OVCAR5 cancer cell line. So they then characterize the exosomes that are released from this cancer cell line. A very typical image of a TEM showing you the morphology of what this exosome looks like right here, below 100 nanometers. And their nanoparticle tracking analysis showing us the modal size of around 81 nanometers, indeed indicating that they are exosomal sized. So here is Western blot indicating the molecular characterization of the exosomes. You can see very clearly that the quite classic exosome markers, TSG101, ALIX, and CD63 are indeed present in their exosome population sample. However, the TC, it's devoid of the cellular contamination marker, the GRP94. So they next determined whether the exosomes carried nuclear proteins, and they performed protein mass spectrometry on the exosomes to do this. They ended up finding around 12.5% of the total chromosome of the total protein was in fact from nuclear proteins or chromosomal protein, and they found that the actual count was around 201 distinct proteins were from nuclear-associated proteins, and 17 distinct proteins were chromosomal in nature. They next looked at the copy number variants of the DNA that's packaged into these exosomes compared to the donor cell. They used the whole genome sequencing, and they used a plot using circus plot. So I can get into the details of what this plot indicates, but it's quite confusing to see. Overall, the cellular DNA outside here, indicated in light blue, is the cellular DNA, and the red here in orange is the exosomal DNA. The green bars indicate insertion, and the red bars indicate deletion, and the depth of the bars indicate the degree of copy number alteration. So overall, their finding was that the copy number variance was quite similar between the exosomal DNA and the donor cell DNA, suggesting that the DNA sorted into exosomes quite uniformly represents the entire genome of the cell. So in the literature, no one really looks at how much of the exosomal population, what fraction of the exosomal population actually has genomic DNA. So this is what they did. They determined if there was any different subfractions within the exosomal population that actually contain DNA. So in this flow cytometry data, you see that each dot is an indication of a single exosome stained in cell mask green. And within the red gate, the red dots indicate DNA positive exosomes. And you can see that already that a clear fraction of the exosome population contain DNA, and not all exosomes contain DNA. So this is quite interesting. They next looked at determining whether lamin AC, a protein which is a nuclear envelope protein, is also present in exosomes. So this flow data here on the left indicates the total exosome number, each dot, and this gating here within the blue gate indicates the lamin AC nuclear envelope positive protein positive exosomes. So you can see already that the lamin AC positive exosomes are only a very small fraction, around 3.5% of the total exosome number. On the right here is a flow data indicating each dot indicating DNA positive exosome, and within the teal gating indicates the DNA positive as well as lamin AC nuclear envelope protein positive exosomes, accounting for around 60% of the total DNA positive exosome. So this immediately tells us that although the lamin AC nuclear envelope protein is quite low overall, if you look at, they are actually they are highly concentrated amongst exosomes that are also DNA positive. They also looked at other nuclear proteins, although the data wasn't shown. They indicated that DNA positive exosomes also contain other nuclear proteins, such as Nesperin-2 and lamin B1. So their next part of the result was investigating the relationship between the micronuclei and DNA positive exosomes, which I'll be calling nuclear exosomes from now on, NXO. So here is, they looked to determine the percentage of micronuclear positive cells. So they used the ovarian cancer 5 cell line. They also used a primary cell, which is a healthy fallopian tube epithelial cell. They used this one because they mentioned that this cancer cell line is a direct derivation from this healthy cell here. So here is a immunofluorescence image of the healthy FTE epithelial cell. This red indicates the lamin AC nuclear envelope protein. The nuclear content is stained with DAPI, and here is emerging. And again, this is the cancer cell line equivalent. And you can see in this magnification panel here that here is a depiction of that micronuclei. And you can see again, within that micronuclei, you see positive nuclear content. And again, the merge here. So overall, they quantified this, and they found that the micronuclei positive cells within the healthy fallopian tube epithelial cells was around 1%, whereas in ovarian 5 cancer cell line, 4% of the cells accounted for micronuclei positive cells. So more genomic instability in the ovarian cancer cell. So they next determined if cancer cells release more nuclear exosomes than normal cells. So here is, okay, hold on. Yep, so this is a total each dot represents exosomes that are released from the healthy FT cell. You can see that within the gating is a DNA positive cell. So you can see again quite clearly that there is very minor release of the nuclear exosomes. Compared to that, if you look at the cancer cell line, compared to the total exosome, within the gating, the red dots indicate the nuclear positive, the DNA positive exosomes. There is clearly a higher release of nuclear exosomes compared to the healthy control. So again, the quantification, we see that the ovarian 5 cancer cell line release significantly higher populations of NXO, around 8.3%, compared to the normal cell line, which is around 0.1% of the total population of released exosome number. And they also tested the ovarian cancer 8 cell line here as well, which showed no difference. So they next determined if the micronuclei can be induced by the addition of genotoxic drugs such as topotecan or olaparib. So here is that data. So here the gray bar indicates the ovarian 5 cancer cell line, and the black bar indicates the ovarian 8 cancer cell line. You can see quite clearly that the total micronuclei positive cells was significantly increased upon addition of the drugs topotecan and olaparib compared to the control DMSO. So they next looked at whether this led to a subsequent increase in the release of the nuclear exosomes. So again, here's the control DMSO, and again, this is the olaparib and topotecan treated groups. You can see very clearly that there was significant increase in the released increased amount of nuclear exosomes in both the ovarian cancer 5 cell line as well as the ovarian 8 cancer cell line with both genotoxic drugs compared to the control. So their third results looks at the micronuclei, whether micronuclei interacts with exosome-associated proteins within the cell. So to determine if the micronuclei and the nuclear exosomes harbor overlapping protein content, first of all, they looked at they isolated out the micronuclei from the cancer ovarian 5 cancer cell line. They isolated out via using sucrose gradient ultracentrifugation. And the fractions that were enriched for micronuclei were submitted for protein mass spec. They then compared the relative nuclear protein abundance in each of the micronuclei compared to the nuclear exosomes. So this data here, the blue bar indicates the protein quantity in the micronucleus compared to the red bar, which indicates the protein quantity in the exosomes for each of these different types of proteins. The green bars indicate the proteins that are chromosomal or nuclear in nature, and you can see already that there is quite a bit of overlap between both the exosomes, and the micronucleus. So this Venn diagram indicates that actually around 127 proteins were overlapping, and this included the nuclear proteins lamin AC, as well as the histone 2B, among others. They next determine whether micronuclei and exosome-associated proteins interact with cells. So they performed confocal imaging of the exosome-associated proteins, CD63 and CD9, which are very classic markers of exosome. So here, the lamin AC nuclear envelope protein is labeled with M cherry, red. The nuclei is stained with DAPI, and the CD63 and CD9 are labeled with immunofluorescent antibodies, which in this case is green. So you can see already that the CD63 and CD9 are co-localizing with nuclear envelope, which is with the nuclear envelope of the micronuclei indicated here, and even within the micronuclei, as indicated here. So they next looked at the exact mechanism behind nuclear content loading into the exosomes. So a bit of background, the micronuclei are unstable and they tend to collapse naturally. The micronuclei, okay, so here is a depiction of a TEM indicating that collapsation of the micronuclei and possible extrusion of that content, of the micronuclei content outside into the cytoplasm. So yeah, upon its collapse, the contents of the micronuclei, including the genomic DNA, are released into the cell cytoplasm. So they questioned, can the collapse of micronuclei induce loading of the micronuclei content into exosomes? So they hypothesized collapsing micronuclei directly interacts with the molecular machinery of exosome biogenesis. So here, the immunofluorescent imaging shows that they observed the tetraspan in CD9 as an example, but they also showed this with CD63. CD9 is a marker of multivesicular body, and it often they show that it often surrounds the micronuclei that are undergoing partial nuclear envelope collapse or complete nuclear envelope collapse. Furthermore, the TEM revealed that multivesicular bodies and early endosomes were in close proximity to the micronuclei. For example, here, the magnified version shows a very clear multivesicular body in close proximity with the breaking down of the micronuclei here. And also the early endosome was in close proximity with the already collapsed micronuclei. So further, confocal analysis showed that intact and collapsed micronuclei were both positive for EEA1, which is an early endosome marker, indicated here. So you see that the green labeled EEA1 is co-localizing with the nuclear envelope protein that has been partially or fully collapsed in the micronuclei. But interestingly, the micronuclei contained less LMAP1, which is a late endosomal lysosome marker. The data wasn't shown, however. So these results overall suggest that genomic DNA can be secreted in an exosome-dependent manner in which the collapsing micronuclei interacts with the endosomal system. So lastly, they wanted to determine whether a specific tetraspanin CD63 is a necessary component for loading micronuclear content into the endosomal system and ultimately into the released nucleoexosomes. So what they did here was they performed a knockdown assay using short hairpin RNA against the CD63. And here is a control without that knockdown. You see that the nuclear exosomes that are released are much higher compared to when CD63 was knocked down in the cell, showing a much significantly reduced release of nuclear exosomes. So here is that quantification. Compared to the control, we see a substantially lower amount of nuclear exosome release. So this indicates that CD63 does play an important role in nuclear content sorting into the nuclear exosome. So I'm just going to read a part of the article, which I didn't include in my presentation. So CD63 can create a complex. They found that the CD63 does create a complex with the genomic DNA and also the nuclear protein histone 2b, H2b, and this may be important for DNA loading into exosomes. And they did this using immunoprecipitation assays. So in summary, around 12.5% of proteins in ovarian 5 cancer cell line, exosomes represent nuclear proteins. The copy number of the DNA sorted into exosomes well represents the donor cell genome. Not all exosomes contain DNA. In fact, a fraction of that is associated with nuclear DNA. And cancer cells have more micronuclei than normal cells, and subsequently, this may result in increased release of nuclear exosomes. The unstable collapsing micronuclei potentially interacts with the early endosomes, and the genomic DNA that spills out from the micronuclei is a potential source of DNA sorting into the nuclear exosomes. Finally, the CD63 was an important component that had a role in nuclear sorting into the nuclear exosomes. So the conclusion was simple. Overall, the study provides the foundation for understanding the cellular mechanisms of nuclear content loading into exosomes. So yeah, that's it. Thank you very much. Any questions?
Oh, thank you, Matt. The chat was really quiet, no questions there, but I have a question. I didn't get why micronuclei tend to collapse.
I don't know as well, sorry. It just said that micronuclei are around 1/4 the size of the nucleus, and they naturally just tend to collapse. Maybe it's like a disposal system. I don't know. It's something that I should look up.
Mm-hmm.
Yeah.
So Matt, what size pieces of DNA do the micronuclei contain? Do they have whole chromosomes or just pieces?
They didn't look at fragmentation data, so I don't know what the actual fragment sizes are. There was no bioanalyzer or tape session data included in this paper. They just did a whole genome whole genome sequencing, which doesn't tell the actual fragment size or intactness. So. But it was quite interesting because it does provide an idea of how DNA can be packaged, which was which is very, very missing in the current literature. We don't know how DNA is packaged at all. However, I think that the micronuclei are not the only mechanism, because we do see in other non-cancer cell-derived exosomes that they do contain quite a substantial amount of DNA, which the micronuclei can't account for. So, yeah, this is just one of, I think, many different mechanisms that may introduce DNA into exosomes or EVs.
Any other questions?
I just want to comment on that flow cytometry. So typically, a typical flow cytometer can't do this sort of work with sorting vesicles if those dots are individual vesicles at the level that's shown here. So they have someone who is exceptionally good at vesicle flow cytometry,
Yeah.
or they have an exceptional machine, or both. That's certainly not something that I think we could do here. Certainly our experiments with trying to look at exosome-sized vesicles are very challenging.
Yeah, yeah.
And do you know what machine they used?
I can look it up.
That'd be cool. Because they there was a presentation at the ICEV meeting in Japan this time last year, where they're using a dedicated nano flow cytometer, the nano FCM.
Right.
And they were able to very nicely quantitate DNA on inside and outside of vesicles through DNase treatment. And it was really nice. It was very nice data. But yeah, most of these ones with flow tend to use a dedicated machine with very, very specific settings. And the discussions here locally have been, yeah, it's too hard. It's too hard to change the settings so much in between users.
Right.
But I don't think we'll be using flow anytime soon.
No, and I mean, the point that Cherie makes there about inside and outside of the vesicles, you know, even when they're using protein markers, were these vesicles permeabilized, or is this just stuff that's on the outside of the vesicle?
I don't know. They didn't mention it. But I did find the machine. I think it's Amnis Image Stream Mark II Analyzer. Is that it? Is that a flow name?
Yeah, it's like $1 million flow.
Okay.
Like, yeah. That's the sort of flow cytometer that takes pictures of each vesicle as it's passing, if I remember right, Colin. Is that correct?
Yeah, 12 different channels if any of them trigger.
Right.
Yeah. We shouldn't even dream about having a machine like that in New Zealand.
We can dream, Larry.
We have a question in the chat from Sandy. Do the other mechanisms of inclusion of DNA into exosomes show whether the DNA is from micronuclei or micro or macronuclei? Can you tell whether the DNA content in vesicles are from macro versus micronuclei?
Okay, hold on. So she wants. I can't see the chat. So whether how can we decipher whether the DNA in the exosomes are from micro compared to nuclear from the actual nucleus? Is that what the question is?
Yeah. Micro versus macro.
I don't think you can, because I mean, what they've done here is just looking at co-localization studies, and they've looked at proteomic studies as well, looking at the nuclear and chromosomal DNA. Unless they find different proteins associated with the actual macro nucleus compared to the micronuclei, and also if they can pinpoint that protein out also inside the exosomes, unless they do that, I don't think they can actually decipher it. I think
So it could be the imaging supports their conclusion, but it's still unclear where the actual DNA came from.
Yeah, at best it's like just co-localization studies that they've did, and I think like when I read this, many of the part was at best assumptions, and just basing off that assumption they just carried on. So I don't know if they can yet decipher whether it's coming from the actual nucleus or the micronuclei.
Do we have other questions from Matt?
Okay. So next week we have Debina presenting, and if anyone would like to volunteer to do the Q&A or to chair the session, that would be wonderful. Just email Anastasia and she can help organize that. And also if anyone would like to volunteer to present after next week, that would be great.
Yeah, we will really appreciate your input.
Okay, no immediate volunteers, but we'll hope for some in the next week. And if no one has any other comments, I think we can close the session.
Thank you, Matt.
Thank you very much.
Thanks, Matt.
Thank
Thanks,
you.
Matt.